Okay, so this is the last lab for the semester for you guys. So we are going to cover 4 chapters, 20 chapters 20 to 23. But basically, this is a sensory nervous system. So we're going to look at sensory receptors, taste and smell briefly, and then eye and vision, and then hearing, ear, hearing, and balance, right? So I'm not going to go over every detail because most of it is in the book, in your manual. I'm just going to give you a brief introduction or a gist of things and the figures you need to worry about. And then I'm going to show you a couple of dissection and cranial nerves affecting these sensory areas and so on. So the rest of it you're going to study from your manual. Okay. Anyway, so in the lecture today, I think I was talking about how under the sensory nervous system or senses, we have two major or general divisions. One is your general senses and the other is the special senses. And so the general senses are, of course, like your pain, touch, pressure, temperature, and so on. Special senses are these are controlled by specialized areas using specialized neuronal pathways like your smell or olfaction, hearing, vision, and so on, right? And also you should remember all of this is made possible because of sensory receptors. They sense all these changes in the environment, whether it is chemicals that you taste or those kinds of things. But these receptors are not uniformly distributed throughout your body, and that is called punctuate distribution. Okay. The other thing is you need to know about what a stimulus is, right? So when there is a temperature change, you feel warmth or cold, right? So that is a stimulus. And so those kinds of stimuli are labeled or identified as modalities. Okay. Now, as we were talking about in the lecture section this morning, the sensory receptors are varied. So you can have mechanoreceptors that are, the stimuli are basically mechanical. Then you can have chemoreceptors, you can have photoreceptors, thermoreceptors, and so on. So there are many receptors depending on what environment they are surrounded by, and also what kind of things they detect. So all those things you have to remember. So in the touch receptors, again, you want to remember from your integumentary system lab and lecture, those little receptors or different types of receptors that are present in the epidermis of your skin, like your Meissner corpuscles or Pacinian corpuscles or Merkel bodies and so on and so forth. You can look at the pictures that we studied there. The histology, of course, is given in your book, but I'm not going to ask you questions using the histology figures, rest assured. Then you can actually check for the receptors by many tests, and some of which we can do in the class tomorrow, like your 2-point discrimination, right? So you basically put like a, what is it called? Compass, right? So you can use a 2-point discriminator or a two-pronged compass or something, and then you can basically touch 2 points of the same tissue, right? But you're going to the person you're testing it on has to have their eyes closed, right? And then you can actually bring the 2 prongs together, and then you want to test at which point, even though you're touching 2 points of the skin, at which point they can still discriminate that as 2 points of stimuli rather than 1 point, right? That tells you how sensitive their receptors are. And then warm and cold, you can do it on yourself, or you can test how easily when a person is not looking at it, how can they say, oh, this is warmer to touch or this is colder to touch, that sort of thing. So there are many tests that you can actually tell. You can do it on yourself or you can do it on another person in order to see how sensitive and how functional their receptors are, right? So like if I'm not looking and then you touch me, if you're touching lightly, am I able to say, oh, okay, I'd move my hand or jerk because I'm not expecting it. And though if I'm not really sensitive to that, you can put your hand on me, I'm still not going to feel it, right? That tells something is wrong with my sensory receptors. um, Same way you can do a test on proprioception. Again, like, the understanding by the brain of all the parts of your body, where they are located, right? So when you're putting your hand on your desk, so suddenly your hand was here, but then if you put it on a desk, now your brain knows that your hand is on the desk, right? And it understands that the position has changed and so on. So all of those things are that's basically covering your that covers your chapter 20. So it's pretty easy. uh, and then let's move to chapter 21, that is your taste and smell. So for the taste, of course, you need your tongue, your taste buds that are there. We'll learn more about the taste buds and so on in your digestive system in the next semester. But right now we are looking at it from a neurological angle. So the sense of taste is also called gustation, right? And then sense of taste, of course, smell, of course, it is olfaction, right? And so you need to know about the, like I said, neurological aspect of it. So which cranial nerve is controlling or sending signals or receiving signals from your nose, for a smell, and from your tongue for the taste. Okay. And in the case of taste, you want to understand what is happening in the tongue. I'm going to show you in the dissection part in anatomy. Mm, and if you want, you can also do tests, right? So you just blot your tongue dry, right, with a paper towel or something like that, and then you close your eyes and then ask your friend or your mate to put like a crystal of salt or sugar, and you should be able to immediately detect it. You may think this is very silly, but there are people when they're they have a nerve injury or something like that, they can't taste. Okay. And more recently, you may have heard that some people in a subset of people who are affected by coronavirus, this COVID-19, they were temporarily, many people lost their sense of smell and taste, and that's what would be, right? Although in many cases they said that a lot of people, whatever they ate, whether it was a hamburger or a mango or something like that, even though these are things that have specific smell and taste, they smelled and tasted gasoline. So you don't know what's going on in the wiring, right, because of the viral infection. So those kinds of things are real, right? And so sometimes we may not have experienced it. So for us, oh, somebody putting a crystal of sugar is a big deal, but that is a big deal for people who can't distinguish between different tastes. So there are different types of tastes, as you know. Some salty, right? Sweet, sugary, and then hot, like chili pepper or something. Then you can have what is called umami. It's like this tart sort of taste, and then you can also taste acidic, right? If you just want to put like a little drop of vinegar, it's basically acetic acid, so it's kind of so it kind of smell, right? It is taste, right? So you can feel that. And then of course there is bitter. So there are many tastes our tongue is able to identify, and this sense of taste is extremely important. And we are given that sense, or we have evolved to have those sense, mainly because we can distinguish harmful substances from things that are safe to eat. right? So imagine by some kind of a developmental problem or genetic problem or some kind of an accident or something, if you lose the sense of taste, you're not, you can be given, you'll be eating poisonous things and you won't know, you can't stop it yourself, and that's very injurious to your health, right? And so sense of taste is very important. Same thing with the sense of smell also. So um, in the case of olfactory system, smelling ability, of course you have olfactory nerve fibers that is involved, right? And then you have cribriform plate, I hope you remember from your skeletal system, olfactory bulb, olfactory tract and everything. So you are supposed to be able to identify these things from this picture in 21.3 on your manual, right? Again, I'm going to show that also as your from your dissection. Let me switch to that now. All right, so let us start with taste here, right? So this is the inferior, we are looking at the brain from inferior angle, right? So what you want to identify here for the taste is that is your facial nerve, or basically as a facial nerve, also called cranial nerve 7, right? So that is important, that innervates your sense of taste. Then you have a vagus nerve, right? Vagus nerve, which is very, very important. Cranial nerve, it's also a very important part of your parasympathetic nervous system. It is also involved in your digestive system, and then regulating your heartbeat and everything, right? That is cranial nerve 10. Then you have glossopharyngeal, which is 9. So all three of them are important for your sense of taste. Then we are also going to look at the tongue, right? So we are looking from above, from superior view. And so you can see the head, top of the head of this person. And so if I'm actually dissecting, you basically end up seeing the lower set of your teeth and then the tongue, right? And let's see what we can, so that is called the sulcus of the tongue. And so you can see the lingual nerve there, not very clearly, but that is a lingual nerve, but that is important in your sensory function. And then chorda tympani, and then dorsum of the tongue, right? And then glossopharyngeal nerve, that you can see also there. That's a glossopharyngeal nerve, the ninth cranial nerve. And then, so that is also another view of the glossopharyngeal. Valate papilla, that is basically part of your taste buds. Foramen cecum of the tongue, terminal sulcus, and the lingual tonsil, right? So we have that lingual tonsil at the end of your mouth, right? So then now let's look at smell, right? From the inferior of the brain. Once again, the same brain, but we are going to not look at here like here, right? Your olfactory bulb, right? And the olfactory tract. So that is it's coming from the base of the brain. Now let's look at the nasal cavity, right? So we've dissected laterally. And so you start to see your olfactory mucosa, and then you start to see your olfactory nerve there. So that is your olfactory nerve, and then the branches are coming here like that. So beautifully arranged, right? And then you have your olfactory bulb again, and then the olfactory tract. And so that is how it is arranged, right? Okay, so let's move on to chapter 22, and that's eye and vision, right? So some of you may have had a course in eye anatomy, because it's a pretty common biological concept taught in even some high schools. But that's even if you have not had that, that's fine. We are going to learn this. And so generally what I want you to focus on are pictures 22.1, 22.2, and so on. And so in 22.1, you get a general idea of what to look for in the eye. So you can just, without dissecting and everything, you can actually see the eyebrow, the eyelid, the pupil, that's the center black thing surrounded by a colored area, which is your iris. And then the white part of your eye is called sclera. And then you can also see the lacrimal coruncle, and then conjunctiva, and so on. And then, of course, your eyelashes. These are all commonly seen, right? But then from this first paragraph, you have all these parts of the eye, including like upper eyelid, eyelashes, and all those things. And then so you need to learn all of those things. And then in your figure 22.2, it actually lists or shows, highlights the muscle, extrinsic muscles and the intrinsic muscles that are important for movement of your eye, right? And so you need to know. So you have your optic nerve there, for example. That's your optic nerve coming from there, right? And then you have your levator, palpebrae, lateral rectus, superior rectus, and so on, right? And they are going to be involved. These muscles are going to be involved in how you are moving your muscle eye. Like, are you closing, are you rotating your eyes this way, that way, or this way, like that, right? A lot of those eye movements, each of these eye movements is dependent on the function of one muscle or a bunch of muscles working together. And so you want to learn all of these muscles. They're also put in this table. Okay. Then you have another function of the eye, and that is tear production. And for that, you have something called lacrimal apparatus. And basically, you have a gland called lacrimal gland inside there, right? I think we also looked at it in your skeletal system part of it. It's like right there inside, right? And there, so the eye actually is in that orb, right, inside. And that orb, so basically it is it's a bulb, and then it kind of cones inside, right, inside the socket. And then so here we have the lacrimal gland, and then it produces the tears which come through the lacrimal duct. And then it actually, so that's why a lot of times when we cry, right, excessively, or when we are sobbing and stuff, we are also producing snot, because it actually has a connection to the nasolacrimal duct. And so both of them get stimulated, and so you can have that when we are emotionally you know we are filled with emotion, whether it is joy or sadness, or sometimes even when you're just cutting onions, right? Either way, those ducts are basically, or the glands are basically activated, and then they produce these secretions. Okay, then there is an interior dissection of the eye, and that actually shows you the interior parts of the eye, like your cornea, and the other kinds of muscles that are present there. And then anterior chamber, ciliary, aqueous humor, and vitreous humor, and so on, right? So, and then where the pupil is located, and so on, right? So you want to learn all of those parts again, and then the retina, of course, which is the place where you uh, that is where that's the most important part of your vision. And then you have your retina is the that converts light energy into nerve impulses. And then you also have bipolar neurons there at the retina, right? And it because the retina is composed of 3 layers. You have a bipolar layer, photoreceptive layer, and ganglionic layer. And the photoreceptor layer is the one that is composed of rods and cones. So they're important for capturing light versus dark, and that's how we actually recognize objects and stuff. And then the cones are important for color vision also. So we can tell I'm wearing red now versus my nail polish is pink, right? So those differences we can say because we have cones in our eye, cone cells. And then you also want to understand what a macula is and the fovea centralis. Fovea centralis is when you actually look at the macula, which is in the book 22:9, right? And you can see the macula, that is this region next to the yellow, right? That is, and then you have this blind spot, that is your fovea centralis, right? And that yellow thing was actually the optic disc. And so that is very important for focusing and so on. All of those things you need to learn, all the parts of the eye. So basically we are talking about eye compartments and ciliary body in 22:5, and then sagittal section of the eye with layers. I'll also bring a model tomorrow to the lab. That is in 22:6. And then you want to understand or be able to identify the retina, the bipolar layer, ganglia, nuclear, and so on from 22:8, and then 22:9, where macula, optic disc, and fovea centralis. All of these things you should be able to understand. Okay. I can bring some charts for astigmatism and color blindness, but those are you need to know what they are, and so you know how to do the test in order for you to focus properly and not have blurred vision, or when something is put in certain colors, that you can distinguish it as, okay, this is red, this is green, because many people who have color blindness cannot make that distinction. Color blindness is a recessive X-linked disease. That means mainly it will, well, mostly it affects males, because they have only one X chromosome, and if it is defective, they'll get the disease. Females, unless they are homozygous for the color blindness, they usually don't have, they're just carriers. So that concludes your eye, but then I'm going to go ahead and show you the dissection for that. Let's start with the inferior brain here. Once again, we are looking at the same, but then now you can see we looked at this part for olfactory, right? Now you can see that's the optic nerve. That is your cranial nerve 2, and that is your optic chiasma, that X-shaped thing, right? That is the optic tract, okay? And let's go to eye lateral, right? And first let's look at so this is the very beautifully depicted eye there. That's your iris and that's your pupil. So okay, now we have dissected the eye, and let's look at the parts here. Okay, let's start with this is your optic nerve coming from the brain, right? Optic disc is here. Posterior cavity, that's the choroid. Retina, right? That's the retina. Sclera. And then you have your ora serrata, a ciliary body. That's the ciliary body. And then suspensory ligament. This is very important. That's the suspensory ligament of the lens. That is your lens, right? And the lens becomes stiffer and stiffer as we age. So if you guys are all very young, and so what you can read, like, if you hold your book here and read without glasses, and like 8 or 10 centimeters from your face, you can read it very well. But then for me, I can't read anything without my reading glasses, right? If you put it here, if I have to keep it like, sometimes 50 centimeters away from me in order for me to read anything. That's because my lens has become more stiff and stiff, and so its ability to focus is lost as we age, right? Anterior chamber, cornea, right? That's the front part, anterior cavity, and then the posterior chamber. Okay, from the orbit, right? So let's dissect. So that is how the eye is arranged inside the socket, in the orb, in the skull. And so you can see how it is arranged. You already start to see the extrinsic muscles and so on. And then let's start to look at the So that's your ciliary ganglion, and that's your optic nerve, right? And then you have short ciliary nerve there. That's the eye, naturally, pupil and your iris there. And so now you can see that's the sclera, suspensory ligament, lens, and cornea, right? And so those are the things you need to, that are visible from a dissection like this. Okay, lastly, chapter 23, we are going to the ear, hearing, and balance. So that is your last of the senses and last of the lab material. So hearing and balance, they're mechanoreceptors because they're going to detect vibrations, right? In the case of eye vision, we used photoreceptors. In the case of ear, we are going to depend on the mechanoreceptors. And then so you need to learn a little bit about the sound. In I, we looked at a little bit, or you're supposed to know a little bit about light, right? So how we light and dark, and then light travels in waves, and it's an energy, and so on and so forth. And then how the eye accommodates and all that to light versus dark. But then in the case of ear, we are looking at sound, which is also travels as waves, and so you need to know about the sound amplitude, wavelength, and frequency. And the amplitude of the sound. Amplitude basically is, if it is a wave, it goes, like that, like sigmoidal, right? And so how much it goes, crest and trough, right? So that how much it goes, that is your amplitude, size of the wave, right? And the unit we measure that is called decibels. And frequency, of course, how often you make the sound travels within a given unit of time. That measurement is usually in hertz. And then balance is dependent on the receptors that are in your inner ear, right? Inner ear, and also visual cues, right? Based on what you see, you can perceive the position and so on. And that is basically your proprioception. Then you have balance in the inner ear. Inner ear actually is very important. It can do both static balance or dynamic or kinetic balance. Okay, this is why if you have inner ear infection, really severe infection, you can't stand straight, or you can't. It's kind of weird, right? To think of, well, I have an infection in the ear, and why can't I stand straight, or why do I keep falling? Because inner ear is very, very important. If it is filled with fluid or something like that, you're immediately, these receptors are going to be messed up, and you're going to lose your balance. Okay. Then we go into the anatomy of the ear. And so you want to understand the external ear. Then you have your tympanic membrane, or the eardrum, right? That's where the vibration is first going to be perceived. Then you have, so external ear, then you have your middle ear. Then you have there you have your tympanic cavity, and then the ossicles. These are the ossicles, if you remember from your skeletal system, those are the smallest bones in our body, right? Those three bones are going to be there. And then you have an auditory tube, and then which attaches to the inner ear. And the inner ear is the one that has the cochlea, that spiral thing, and the vestibule, and the semicircular canals, right? And so all of this is depicted in this picture 23-2, right? And so the ear ossicles, right, they are actually present here in this place: malleus, incus, and stapes, right? And then this is your cochlea, right, the spiral area. And then those little handle like things, those are your semicircular canals. And then the passageway to the cochlea, right, that is your vestibule. And then in the auricle, the external ear, this is called auricle, or also pinna, right? You have to remember that's made of your connective tissue, right? Then you can also call it a earlobe or whatever. And then you have the external ear divided from the middle ear by the tympanic membrane, eardrum. So that's why with very, very high frequency of sound, or very high amplitude of the sound, or otherwise infection or some kind of autoimmune reaction or a mechanical injury, you can, if you puncture your that drum, eardrum, you're basically going to lose your hearing. It's very important. Then you want to look at the middle ear. Like I said, you have the tympanic cavity, and then you have the ossicles there, and then your auditory tube, and then from the middle ear to your pharynx, right? From the middle ear, you go up to inside, right? And then to the pharynx, that area is called your upper respiratory tube, also called eustachian canal or eustachian tube. That is actually connecting your inner pharynx, inner organ, to the external environment, right? So that is eustachian canal is extremely important, and that is one of the reasons when you have sometimes when you have, like, say, a cold where your nasal cavity is blocked, your ear also gets blocked because of the connection by the eustachian canal. So the ossicles, malleus, incus, and stapes, they are present in the middle ear, and they basically sense they're going to vibrate. If your tympanic membrane senses the vibration, it is any sound, right? And that is going to be transmitted to the ossicles, and they are going to vibrate. And so there is a whole lot of vibration moving from one place to the other, which results in our perception of sound. Right? And then in the inner ear, you have your bony labyrinth, and then you have a solution called or fluid called perilymph, right? And then you have your membranous labyrinth. And then in the membranous labyrinth, of course, you have the endolymph, another fluid, right? And those two, perilymph and endolymph, they're very important, especially your endolymph. That has to be in the right place and in the right quantity for us to be able to hear properly and to keep our balance. All right. And so the middle ear, right, that is depicted here, right, with your ossicles, very nicely put. And then you have the inner ear here, right, the cochlea, vestibule, and the semicircular canals, and the fluid thereof, and so on. Okay. And so you can look at the cochlear structure in more detail in this picture 23:6, and you can look at the oval window and round window. Those are all parts of the inner ear. And then there are also your scala vestibuli, scala media, and other parts of the cochlea. And again, same thing with your vestibule, and then with the semicircular canals. All of these parts are very important because they are contributing to your ability to transmit the sound and then sense it, and so on, right? And then basically, yeah, and then you're in how do you interpret sound, right? And that is given in this figure 23:8. How it starts with the sensory axons from the cochlear ganglion. They go to the brain stem, and then processed in the brain, and then you perceive it as Dr. B talking versus my mother talking, or my father talking, or my friend talking. All those voices, you are familiar with so many voices, but you're able to identify one voice from the other, and that is because of the processing centers in the brain, right? And so basically in your inferior colliculus, and then another place called medial geniculate nucleus, right? And all of both of those, that's in the hypothalamus, that's in the thalamic area of the brain. And then the thalamic area gets all that information, and then it projects to a part in the cortex called auditory cortex, right? And auditory cortex are they're going to then bring the thing back to the inner ear, from neurons emanating from there to the inner ear, and that's how you perceive the sound. And then your inner ear muscles are also going to be involved in this process, right? There are many tests that you can do in order to test a person's ability to hear and respond. And there are because deafness is a very common disease. People can go deaf for many reasons. One of the major reasons for deafness in today's world is people listening to very loud music all the time, right? And that basically wears off your thing and your sensitivity. And then you can actually do audiometer tests if you want to do it online. There are many, you can just type in audiometer and then they give you sounds and everything. And so you can actually see there is a range of sound that is considered normal, but then when it is played on your computer, if you're not able to hear it, or if you have to increase the volume very much, that means you're going deaf, right? And anyway, so there are two kinds of deafness. One is called conduction deafness, and the other one is called sensor sensory neural deafness. In conduction deafness, the outer ear or middle ear, um, the environmental disturbances or whatever, right? Those are not processed or not able to be perceived in the outer or the inner ear, and therefore they don't even reach your cochlea, right? And that, like I said before, the tympanic membrane injury can also block the thing. Sensory neural is in the cochlea, in the, in the middle ear, where, oh, sorry, in the inner ear, where you can actually damage the cochlea, or the cochlear neurons have become injured or something like that, or developmental problems, right? All of those things. So I don't know how many of you are familiar with it, but in some children, there is actually, when they have developmental problem or some kind of a congenital problem, and so they can't hear. There is a window within 12 to 18 months, I think, of after birth. And within that window, actually, if that is actually identified, you can actually do what is called cochlear implant. And then these children can actually get they can actually start to hear. But the problem is, in many cochlear implantation techniques, you have to do the implantation within that window of time, because after that, when the ear develops, cochlear implant doesn't have any effect, and so they will remain deaf. The other problem that can cause hearing loss in such infants is, if they have some kind of a infection, and so they're administered antibiotics. Some antibiotics are very ototoxic, which means they will affect growing parts inside the ear, specifically those cells. And if that happens to be a cochlear cells, naturally that's very bad. And so that's very common also. Like some antibiotics, like streptomycin or something, they can actually kill the developing neurons there and cause deafness. We'll try and do Weber test tomorrow. That's basically, you strike your tuning fork. There are many tuning forks with the specific frequency of sounds, and so you just strike it and then hold it near your ear and then see if you can hear it, right? And are you hearing it louder in one ear or softer in another ear? And those kinds of tests you can do. Weber test is a very common test in order to understand whether you have deafness or not, or hearing loss or not. Another test is called RIN test. That also you can do, and so we can check those kinds of things. Okay. So let me finish by showing you a dissection for the auditory system. Let's see. Where is it? Yeah. So let's start with the inferior of the brain, same brain. We are going to look at the nerves. Basically, that's your vestibulocochlear nerve. as you see, as the name goes, it's innervating your vestibule and the cochlear areas in the inner ear. That's basically cranial nerve 8. And then let's look at the ear here, and that's your external ear auricle pinna, and that's a external acoustic meatus, temporal bone, that's the temporal lobe of the brain, right, coming in there. That's your tympanic membrane there, right? So that is there. And then you have a malleus, incus, and stapes. Tympanic cavity, that's the middle ear, and that's the auditory tube that's going to go inside like that. And then you have your vestibule of the ear, semicircular canals, cochlea, and then your cranial nerve 8. Okay, I think I'm going to stop with that. And I will see you guys tomorrow in lab.